IB Bio Human Physiology Part 8 is the second of two parts covering the defense against disease, the immune system. In Human Physiology Part 8, Defense Against Disease B, this movie, the focus will be on the defense system that is specific to the pathogen. Humans have immune cells that are primed to track down and remove specific pathogens. It is the specific defense system that forms the basis of our immunities to specific diseases once infected or once injected. The essential idea is the human body has structure and processes that resist the continuous threat of invasion by pathogens. Here is the first of two slides that provide an outline of all movies available for Topic 6, Human Physiology for SL and HL students. Use the outline to find the movie you need for review. This movie is focused here. Here is the second slide of the outline for all movies available for Topic 6, Human Physiology. Defense Against Disease B, Part 8, will look at macrophages, B cells, and T cells. And remember that a pathogen is a disease-producing organism. Stem cells from the bone marrow result in our red blood cells, the platelets, which are involved in blood clotting, and the array of white blood cells that we have to fight infection. The focus of this movie is here, the lymphocytes, also known as B cells and T cells, and these serve to identify specific pathogens, and they clone themselves in great numbers to fight the infection. The B cells and the T cells are the central players in immunity. The B cells and the T cells are the specific cells that protect us against specific pathogens and the substances that those pathogens produce. So immunity is the capacity to resist pathogens that would damage tissues. Immunity allows us to be exposed to pathogens without risk of a larger infection. Most of you have had the chickenpox and are now immune from another infection of the chickenpox. Most of you have had a vaccination for measles, mumps, and rubella, so that you will not experience an infection from these pathogens. Now, just to set the stage for how the specific defense systems work, let's distinguish B cells and T cells. Most of our infections begin in the bloodstream, and that's where the B cells work. It's called the humoral response. The response to the antigen involves antibodies and occurs outside body cells, in other words, in the blood. But once a pathogen infects a body cell, gets inside a cell, the T cells go to work. This is called the cell-mediated response, and the response is directed at body cells that are infected or invaded by the pathogen. So once again, in different words, in the battles, out in the open, the bloodstream, the B cells mount the attack with antibodies. But once the invader sneaks into cells to evade the B cells, the T cells work to sniff out the hiding pathogen. But before we can talk about how the B cells and T cells identify the pathogen, we need to distinguish antigens and antibodies. An antigen is any molecule recognized as foreign that stimulates antibody production. Antigens are just small molecular components of larger pathogens, but it is the molecular components, the antigens, that our B cells and T cells recognize as foreign. B cells make antibodies specific to the antigen. The antibodies bind to the antigen and neutralize the antigen to ultimately neutralize the pathogen. This slide is detailed, but it is terrific to study as a way to understand both the small and big picture aspects of the specific defense systems. I will use it repeatedly in this movie. Pay close attention as I will choose certain sections to emphasize. I will not explain the entire image in one go. The antigen is a molecule on the surface of a pathogen. The antigen is a molecule on the surface of a pathogen recognized directly by B cells and T cells as foreign. B cells recognize the presence of the antigen and are stimulated to divide into many B cells of the same type specific to the antigen called a clone. The B cells then make antibodies specific to the antigen that will neutralize the antigen and thus neutralize the pathogen. In this image, you can see an antibody. It is a Y-shaped protein structure. Antibodies are soluble proteins that bind antigens secreted by B cells that are specific to the antigen. I've shown you this slide before as an outline of blood cells. We're focused here on lymphocytes called B cells that make antibodies. Antibodies are soluble proteins manufactured by B cells, released into the bloodstream, that bind the antigens on infecting pathogens. As lymphocytes, the B cells secrete antibodies into the bloodstream as a defense against pathogens out in the open, outside cells. T cells activate B cells, and the T cells serve as a defense against pathogens that have entered cells. The production of antibodies by lymphocytes in response to particular antigens or pathogens will give us specific immunity. Blood cells, red and white, originate in bone marrow as stem cells, open to differentiating into many different types of blood cells, including B cells and T cells. B cells mature in the bone marrow, while T cells, once formed, mature in the thymus. The B cells and the T cells reside typically in the lymph nodes, the spleen, and in the bloodstream. The bone marrow is the site of blood cell formation, beginning with stem cells that are open to differentiation. Maybe you already see what's happening. The pathogen arrives with its antigens on the surface. It's a challenge to our homeostasis. Our bodies respond to the challenge with cells, macrophages, B cells, and T cells that target the specific pathogen, ready to defend the body's homeostasis. Here's the relevant IB syllabus statement. Outline the principle of challenge and response, clonal selection, and memory cells as the basis of immunity. An antigen challenges, the lymphocytes respond, and we have clonal selection, where an antigen, by binding to specific receptors, selectively activates a tiny fraction of cells from the body's diverse pool of lymphocytes, and they give rise to thousands of cells, including memory cells and antibodies. The antigen challenges, stimulating B cells directly, and if engulfed by macrophages, the macrophage will stimulate helper T cells that help stimulate B cells and cytotoxic T cells. The B cells will clone themselves and make antibodies specific to the antigen to neutralize the pathogen. When B cells are stimulated by the antigen, the B cells clone, divide through mitosis. The huge number of cells are then ready to make antibodies that neutralize the antigen. As well, some of the cloned B cells will become memory cells that reside in our bodies for some time, ready to fend off the pathogen upon a second contact. This is the immunity we get from first infections or from vaccinations. Let's look at some of the detail of how a macrophage, once it has consumed an antigen, stimulates helper T cells and then B cells. The macrophage, seen here, once having engulfed the antigen, presents pieces of the antigen on its membrane surface. Helper T cells, specific to the antigen, are stimulated by the specific molecular shape of the antigen. Once the macrophage, here, and the helper T cell, here, contact each other and bind, the helper T cell is activated. Now the helper T cell activates B cells, and the production of antibodies by B cell lymphocytes in response to the specific antigen that's arrived give us specific immunity. Now, before leaving this slide, I want to just note the MHC surface proteins. These receptors allow cells of the body to recognize each other as self. Macrophages of the body and the lymphocytes communicate with each other using the MHC surface receptors. The helper T cell, specific to the antigen, binds with B cells with receptors specific to the antigen. The B cells become activated and begin secreting antibodies that will bind and neutralize the pathogen. And the B cells, once activated by the helper T cells, will clone themselves into a large colony of cells all ready to fight the specific pathogen. And then some of those cloned cells will reside in the body for long periods of time as memory cells, ready to defend the body against the pathogen upon a second exposure. So this is what's meant by clonal selection. An antigen, by binding to specific receptors on macrophages or lymphocytes, selectively activates a tiny fraction of cells from the body's diverse pool of lymphocytes, and they give rise to thousands of cells, including memory cells. This graph shows antibody production across time, and this blue line follows antibody production through a first exposure and a second exposure to antigen A. Here's antibody production following the first exposure. Antibody numbers rise to a peak approximately 14 days after the exposure. But upon a second exposure here, thousands more antibodies are secreted within 7 days of the second exposure. This results because of the memory cells. As I've been talking a lot about antibodies, let me simply introduce the phrase monoclonal antibodies. Monoclonal antibodies are antibodies of a singular molecular type secreted by B cells in response to a specific antigen. Nothing more than that. HL biology students will look again at monoclonal antibodies later in the course. While there are other T cells, known as cytotoxic T cells, that target infected cells, cells that have a viral infection, and kill those cells, we can hold our focus to helper T cells and B cells, the clone, the antibodies, and memory cells. Now, it's important not to forget what helper T cells do in terms of B cell activation, because it is helper T cells that are infected by the HIV virus. When someone is infected by the HIV virus, their helper T cell numbers decline. And when helper T cell numbers decline, the patient is at risk of other opportunistic infections that they can't fight off because the B cells are not stimulated properly. Once again, the macrophage, once having engulfed the antigen, presents a piece of the antigen on its membrane, and that stimulates the helper T cell. This is known as antigen presentation. Helper T cells secrete a hormone known as a cytokine that stimulates cytotoxic T cells. The cytotoxic T cell will kill body cells that are infected with the specific antigen, a viral antigen. This slide organizes a lot of what I've presented in this movie. It's a specific defense system that employs the lymphocytes of B cells and T cells, as well as macrophages. Spend some time studying this slide. Helper T cells are central to the stimulation of our immune system. Helper T cells stimulate cytotoxic T cells with a hormone known as a cytokine. Helper T cells stimulate B cells directly through antigen presentation and stimulates B cells with cytokines. Here are the next IB syllabus statements. Outline the effects of HIV on the immune system. State reduction of active lymphocytes and reduction in antibody production leading to the development of AIDS. State methods of HIV transmission. HIV is a virus, and remember, antibiotics interfere with bacterial metabolism, but not viral metabolism. Antibiotics do not work on viruses because viruses rely on host cells to multiply. Viruses do not have their own internal metabolism that would be vulnerable to antibiotics. The HIV virus, seen here, infects helper T cells. The helper T cell is the host cell that the HIV virus infects. HIV, the human immunodeficiency virus, infects cells that bear certain receptors such as helper T cells, macrophages, and some B lymphocytes. With the decline in helper T cells, there is real loss of active lymphocytes and a reduction in antibody production. Remember, helper T cells are stimulated directly by macrophages. Helper T cells help stimulate both B cells and cytotoxic T cells. HIV reduces the number of helper T cells, thus the entire immune system is compromised. This graph displays helper T cell concentration across time following HIV infection. You can see the number of helper T cells declines over time following HIV infection. HIV, by infecting helper T cells, compromises the entire immune system. So, can you outline the effects of HIV on the immune system? Can you state that there's a reduction of active lymphocytes and a reduction in antibody production, leading to the development of AIDS, which is acquired immunodeficiency syndrome? Can you state the methods of HIV transmission? Here's how HIV is transmitted: through blood transfusions, blood-contaminated needles, intimate sexual contact, across the placenta from mother to baby, and tears, sweat, mucus, mother's milk also have low concentrations of the HIV virus, but very low concentrations. In the micrograph, we can see an artificially colored purple HIV virus on the surface of a salmon-colored T cell. I've come back to this graph to distinguish HIV from AIDS, acquired immunodeficiency syndrome. AIDS is caused by HIV, but they are not the same. Take a close look. HIV infection takes place here. HIV resides within the helper T cells, causing the helper T cell number to decline over a very long period of time. Only when helper T cell numbers have declined to low values and antibody production against HIV begins to decline does the onset of AIDS occur. The onset of AIDS is marked by opportunistic disease, the arrival of certain types of cancer, sweats, weight loss, and more, a syndrome of effects. The symptoms of acquired immunodeficiency syndrome are susceptible to opportunistic disease, flu, pneumonia, cancers, certain kinds, Kaposi sarcoma, severe weight loss, night sweats, swollen lymph glands. The cause of AIDS is the retrovirus, human immunodeficiency virus, HIV. And that brings us to the end of IB Bio, Topic 6, Human Physiology, Part 8, Defense Against Disease, B. In Topic 11 for HL students, Part 1 would serve all students well for strengthening what I presented here